This is a recorded lecture for Chapter 23, Nutrition, Metabolism, and Body Temperature Regulation. This is this will be a chapter in the A&P 230 section in the second semester. Nutrient is defined as a substance in food that promotes normal growth, maintenance, and repair, and that includes carbohydrates, lipids, and proteins. We have other nutrients like vitamins and minerals. Technically speaking, we need water, but it has no nutritional value in itself. It's a great solute, but it has no nutritional value in itself, but we do need it. This is the mypyramid.gov, which talks about steps to a healthier you and talks about what we need to take in based on USDA recommended amounts. As you can see, the daily exercise and weight control down at the bottom. We need to take in whole grains and whole grain foods at most meals. Should be some plant oils at most meals. Fruits, you can have 2 or 3 servings a day. We have vegetables in abundance. You see that nuts and legumes are 1 to 3 servings a day. We have fish, poultry, and eggs, which can be 0 to 2 servings a day. Dairy or calcium supplement, which could be milk or cheese or those kind of things, 1 to 2 servings a day. And finally, red meat and butter and white rice, white bread, potatoes, pasta, and sweets, we should use very sparingly because they readily go to fat and are not necessarily good for us. Dietary sources include a starch which is complex carbohydrates found in whole grains and vegetables. Also, you find sugars in fruits, sugarcane, sugar beets, honey, and milk. And then there's an insoluble fiber, which is the cellulose in vegetables and provides the roughage, and that includes like the husk of a corn when you eat corn. And then soluble fiber, which is the pectin in apples, citrus fruits, which reduces the blood cholesterol levels. The use of carbohydrates, glucose is a fuel used by cells to manufacture ATP. Neurons and red blood cells rely almost entirely upon glucose to produce ATP. Excess glucose, unfortunately, is converted to glycogen, and then it's stored as fat, or it's ultimately stored as fat if you don't use the glycogen. So it's important that you have a daily exercise plan to use up your glycogen. Dietary requirements of carbohydrates. There's a minimum of 100 grams a day to maintain adequate blood glucose levels, and it's recommended a minimum of 130 grams a day. Recommended intake should equal 45 to 65% of your total caloric intake, and most of those should be complex carbohydrates like in vegetables and such, not simple carbohydrates like sugars. Dietary sources of lipids we're going to talk about are triglycerides and cholesterol. Triglycerides come from saturated fats in meat, dairy products, and tropical oils. They are also unsaturated triglycerides, which are fats we get from seeds and nuts and olive oil and most vegetable oils. These are not so good. These are pretty good. Cholesterol can be found in egg yolk meats, organ meats like heart, liver, kidneys, if you will, shellfish, and milk products. We have essential fatty acids, which include linoleic and linolenic acids, which are found in most vegetable oils. They must be ingested, obviously. And the essential use of lipids in the body is they help absorb fat-soluble vitamins. They're a major fuel for hepatocytes and skeletal muscle. Phospholipids are essential in myelin sheath and also membrane production. So without lipids, you really don't, the oligodendrocytes and the Schwann cells don't have the material needed to provide the myelin sheaths required. Lipids the functions of fatty deposits, adipose tissue, help protect and cushion the body. It insulates it provides an insulating layer of skin, and it's a concentrated source of energy, though it's a complex one that has some drawback when you break it down. The regulatory function of lipids, prostaglandins, include smooth muscle contraction, control of blood pressure and inflammation. The functions of cholesterol is it helps to stabilize membranes, and is a precursor of the bile salts. Also, our steroid hormones, which is pretty darn important when you stop to think about all the hormonal activities that require a lipid foundation. The dietary requirements suggested by the American Heart Association is fat should represent 30% or less of our total caloric intake. Saturated fat should be limited to 10% or less of our total caloric intake, total fat intake, and daily cholesterol intake should be no more than 300 milligrams a day. So that's not very much. Proteins, we have dietary sources of proteins include eggs, milk, fish, and most meats, which contain complete proteins. We also have legumes, nuts, and cereals, which contain incomplete proteins. They lack some of the essential amino acids. Legumes and cereals together contain all of the essential amino acids, and that's important to know. That's how vegetarians, if you will, can survive without eating meat. The use of proteins are the structural materials in keratin, collagen, elastin, and muscle proteins, and they also provide most functional molecules with some substances, including enzymes and some of the hormones. Proteins the use of amino acids in the body. It's an all or none rule. All amino acids need to be present for a protein synthesis process to occur. The adequacy of the caloric intake, protein must be used as fuel, or will be used as fuel if there's insufficient carbohydrate or fat availability. So when people go on starvation diets and they take in no carbohydrates or fats, what happens is your body starts to break down your muscle tissue and the other proteins of the body instead. That's what happens to people that have anorexia nervosa. They start breaking down the muscle tissue. By the way, that includes your heart and your all the other muscles, your smooth muscles of your or other organs, and so that's not a good thing. The nitrogen balance is a state where the rate of protein synthesis is equal to the rate of protein breakdown and loss. The nitrogen balance is positive as synthesis exceeds the breakdown. That's normal in children, by the way, and during tissue repair. The nitrogen balance is negative if the breakdown exceeds the synthesis that occurs in high stress, in burns, in infection, or in any major injury. And then proteins are under hormonal control. The hormonal control include anabolic hormones, which include growth hormone and sex hormones, and then they accelerate protein synthesis. So you kind of have a feel for all the things that proteins do. This looks at the essential amino acids, if you will, and where they come from, and you can follow this in your book. The dietary requirements for proteins, the rule of thumb is that your daily intake should be 0.8 grams per kilogram of body weight. Kilogram is 2.2 pounds, so you could kind of do the math. Another nutrient is vitamins, and they're organic compounds which are crucial in helping the body use our other nutrients. And most of the vitamins function as what are called coenzymes. Vitamins D, B, and K are actually synthesized within the body proper. There are two types of vitamins based on solubility. There's water-soluble vitamins, that would be the B complex and C, which are absorbed with water. B12 absorption requires an intrinsic factor, and then of course they're not stored in the body. They have to be taken in every day. There's also fat-soluble vitamins. They include vitamin A, D, E, and K, which are absorbed with lipid digestion products. They are stored in the body, except for vitamin K, which is produced in the gut by bacteria in the gut. And then vitamins A, C, and E act as antioxidants. Minerals, there are 7 required in moderate amounts. They include calcium, phosphorus, potassium, sulfur, sodium chloride, and magnesium. Others are required in trace amounts. And they work with nutrients to ensure proper body functioning. And their uptake and excretion must be balanced to prevent a toxic overload, because too much of any of this is actually toxic to the body. The next nutrient we're going to talk about is really minerals. And the examples include calcium, phosphorus, magnesium salts, which harden the bone, iron, which is essential for oxygen binding to hemoglobin, iodine, which is necessary for thyroid hormone synthesis, and then sodium chloride, which are the major electrolytes in the body, or in the blood, excuse me. Metabolism is all the biochemical reactions inside the cells which involve the transformation of nutrients. And there are 2 types of reactions. There's an anabolism or anabolistic reaction, and a catabolism, which is a catabolistic reaction. Anabolism is a synthesis where the result is that smaller molecules are made into larger molecules. Catabolism is just the opposite. And I always remember catabolism like cannibalism, where you break down something. It's the hydrolysis of complex structures into simple ones, so it's the breakdown. Anabolism is the buildup, catabolism is the breakdown. Cellular respiration is defined as catabolism of food fuels, and the capture of energy to form ATP in the cells to provide energy for cell activities. Enzymes shift high-energy phosphate groups of ATP to other molecules, and that's called phosphorylation. phosphorylated molecules are activated to perform several cellular functions. The stages of metabolism: there's the processing of nutrients, where digestion, absorption, and transport to tissue. Cellular processing, the cytoplasm, where there's synthesis of lipids, proteins, and glycogen. Or there's catabolism, glycolysis, intermediate molecules. There's also oxidation, and that tends to be in the mitochondria, whereas the breakdown of intermediate molecules into CO2, water, and ATP. And this takes you through those stages. It's a really nice table to go through, and you can see that we'll talk more about this as we move on. We have to go back to talking about redox reactions. We talked about oxidation reduction reactions back in the chemistry chapter 2 section of the A&P book. But oxidation is when there's a gain of oxygen or a loss of hydrogen. So oxidation is when there's a extra oxygen gain or the loss of hydrogen. Oxidation reduction reactions, redox reactions, is where oxidized substances lose the electrons and energy, and reduced substances gain electrons and energy. So it's important to know that. Oxidized substances in a redox reaction actually lose an electron and some energy. And reduced substances in an oxidation reduction reaction, redox reaction, gain electrons and energy. Coenzymes act as hydrogen or electron acceptors, and so nicotinamide adenine dinucleotide NAD and flavin adenine dinucleotide FAD are important coenzymes which act as hydrogen, so NAD and FAD. ATP synthesis, there's two mechanisms. There's a substrate level phosphorylation. There's an oxidative phosphorylation. The substrate level phosphorylation is where high energy phosphate groups are directly transferred from phosphorylated substrates to ATP. This occurs in glycolysis and the Krebs cycle. This is a substrate level phosphorylation. You see an enzyme, there's an ADP, a free phosphate, there's a substrate, and what you're seeing is that there's after the activity, there's a catalysis that comes up here, if you will, and that the product is here, and the result is ATP at the other end. Oxidative phosphorylation is a chemiosmotic process. It couples the movement of substrates across the membrane to create chemical reactions. Oxidation phosphorylation of the mitochondria is carried out by electron transport proteins, and nutrient energy is used to create this positive gradient, this hydrogen gradient across the mitochondrial membrane. At that point, free flowing hydrogen flows through the ATP synthesis, and energy is captured and attaches phosphate groups to ATP, creating ultimately ATP. This talks you through the oxidative phosphorylation, and you can see what we're talking about as we go through. We have carbohydrate metabolism, and that's the oxidation of glucose, where you see this chemical reaction occurring. Ultimately, what happens is C6H12O6 plus 6 oxygens end up with 6 molecules of water, 6 molecules of carbon dioxide, 36 ATPs in the release of heat. Glucose is catabolized in 3 major pathways: glycolysis, the Krebs cycle, and then electron transport chain and oxidative phosphorylation. This takes you through that process, if you will, and talks you through glycolysis, the Krebs cycle, and then electron chain and oxidative phosphorylation. You can see that during glycolysis, each glucose molecule is broken down into 2 molecules of pyruvic acid in cytosol, creating ATP. In the Krebs cycle, the pyruvic acid then enters the mitochondrial matrix, where the Krebs cycle decomposes it to CO2, carbon dioxide. During glycolysis, the Krebs cycle, small amounts of ATP are formed by a substrate level phosphorylation. And finally, energy-rich electrons are picked up by coenzymes that are transferred to the electron transport chain. They're built into the crista membrane of the mitochondria. The electron transport chain carries out oxidative phosphorylation, which accounts for most of the ATP generated by cellular respiration. And we saw that before. Glycolysis is a 10-step pathway. It's anaerobic. It occurs in the cytosol, in the fluid, if you will, inside the cell. And glucose is translated into two pyruvic acid molecules. There are three major phases. There's sugar activation, sugar cleavage, sugar oxidation, and ultimately ATP transformation or ATP formation. The phases of glycolysis, sugar activation is where glucose is phosphorylated by two ATPs to form fructose 1,6 biphosphate. There's then sugar cleavage where the fructose 1,6 biphosphate is split into 3 carbon sugars, dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. Sugar oxidation, ATP formation, the 3 carbon sugars are then oxidized, reducing the NAD we looked at before. The inorganic phosphate groups, PI, are attached to each oxidized fragment, and the result is 4 ATPs, which are formed by substrate-level phosphorylation. And this takes you through all those steps that we just went through. Glycolysis. The final products of glycolysis include 2 pyruvic acids. They're converted to lactic acid if oxygen is not readily available. And they enter the aerobic pathway if oxygen is readily available. So this could be an anaerobic or an aerobic process. We have 2 NADH molecules plus hydrogen, which reduces the NAD, which creates reduced NAD, and the net gain is 2 ATP molecules. The Krebs cycle occurs in the mitochondrial matrix on the cristae. It's fueled by pyruvic and fatty acids. And there's a transitional phase. Each pyruvic acid is converted to acetyl coenzyme A. The decarboxylation is the removal of 1 carbon to produce acetic acid and carbon dioxide. Oxidation occurs where hydrogen is removed from the acetic acid and picked up by the NAD. Acetic acid and coenzyme A form then acetyl coenzyme A. Coenzyme A shuttles acetic acid to an enzyme of the Krebs cycle, and each acetic acid is decarboxylated and oxidized, generating 3 NADH plus a hydrogen, 1 FADH2, 1 CO2, and 1 ATP. A lot of work for 1 ATP, as you can see. Krebs cycle does not directly use oxygen, and the breakdown products of fats and proteins can enter the cycle. The cycle intermediate molecules may be used as building materials for anabolic reactions also. This takes you to the Krebs cycle. It's a very interesting cycle. I had to learn it step by step when I went through the physiology. Electron transport chain and oxidative phosphorylation. This is the part of metabolism that directly uses oxygen as one of its resources. And it's a chain of proteins are bound to metal atoms or cofactors on the inner mitochondrial membrane. And the substrates NADH plus H, hydrogen, and FADH2 deliver hydrogen atoms. Hydrogen atoms are split into hydrogen and electrons. The electrons are shuttled across the inner mitochondrial membrane and losing energy at each step. The released energy is used to pump hydrogen into the inner membrane space. Respiratory enzyme complexes 1, 3, and 4 pump hydrogen into the inner membrane space, like you said. Hydrogen diffuses back to the matrix via ATP synthesis, and ATP synthase uses released energy to make ATP. And this goes through that process, and you see that go through the mitochondrial function. Electron transport chain and oxidative phosphorylation. Electrons are delivered to oxygen, forming oxygen, negative oxygen. Negative oxygen attracts hydrogen to form H2O or water. And I say negative oxygen because it's a negative charge. And this takes you through that process also. We have electronic energy gradient. That's the transfer of energy from NADH plus a hydrogen and FADH2 to oxygen, which releases large amounts of energy. This energy is released in a stepwise manner through the electron transport chain. And it goes through ATP synthase. There are 2 major parts connected by a rod. The rotor is in the inner mitochondrial membrane, and the knob in the matrix. Works like an ion pump in reverse. In that, a rotor is in the membrane, it spins clockwise when hydrogen flows through it, and creates an H+ gradient. A stator anchored in the membrane holds the knob stationary. As the rotor spins, a rod connecting the cylinder rotor and knob also spins, and the protruding stationary knob contains 3 catalytic sites that join inorganic phosphate to ADP to make ATP when the rod is spinning. So it's kind of like this little engine. This takes you to that same process that we just talked about. Glycogenesis and glycogenolysis. Glycogenesis is glycogen formation when glucose supplies exceed the need for ATP synthesis, and it's mostly in the liver and skeletal muscles. So they take glycogenesis as the production of glycogen to use for further energy requirements in the future. Glycogenolysis is where the glycogen breakdown in response to low blood glucose and the need for more energy. This takes you to that process we just talked about. Glyconeogenesis is glucose formation from molecules that are non-carbohydrate, like glycerol and amino acid molecules. Remember that the body can create carbohydrates, can create glucose from non-carbohydrate sources, including fats and amino acids. This occurs mainly in the liver, and it protects against damaging effects of hypoglycemia, so we have another way to do that. Of course, if somebody's on a starvation diet, it also starts to destroy the muscle tissue of their body. Lipid metabolism. Fat catabolism yields 9 kilocalories per gram versus 4 kilocalories per gram of carbohydrates or protein. Most products of fat digestion are transported as chylomicrons and are hydrolyzed by endothelial enzymes into fatty acids and glycerol. Only triglycerides are routinely oxidized for energy. And the 2 building blocks are oxidized separately. There's a glycerol pathway and a fatty acid pathway. The glycerol is converted to glyceraldehyde phosphate and enters that Krebs cycle, and it's equivalent to half a glucose molecule. The fatty acids undergo beta oxidation, which produces 2 carbon acetic fragments, which enter the Krebs cycle. They are reduced coenzymes, which enter the electron transport chain. Again, takes you to that same process. Lipogenesis. Triglyceride synthesis occurs when cellular ATP and glucose levels are high. So there's a production of triglycerides. Glucose is easily converted to fat because of acetylcoenzyme A is an intermediate in glucose catabolism, and it's a starting point for fatty acid synthesis. So it's the coenzyme A that is able to take that glucose and store it as complex carbohydrates, that's a glycogen, or as translated into fat. Lipolysis is the reverse of lipogenesis, and its oxaloacetic acid is necessary for the complete oxidation of fat. Without it, acetylcoenzyme A is converted by ketogenesis to the liver into ketone bodies, and high levels of ketones are not good for the body. It works almost in a destructive way, and it's similar to taking too much alcohol, except there's a whole lot of bad things that can happen. And this takes you to that process that we just talked about. There's a synthesis of structural materials where phospholipids are synthesized for cell membranes of myelin. Cholesterol is synthesized for cell membranes of steroid hormone synthesis. In the liver, the synthesis of transport lipoproteins is accomplished for development of cholesterol and fatty acids. Synthesis of cholesterol from acetylcoenzyme A, and then the use of cholesterol to form those bile salts we need for digestion of fats. Protein metabolism is when dietary protein is excess. Amino acids are oxidized for energy or actually converted also into fat for storage. So too much protein is also not a good thing unless you use it. The oxidation of amino acids, they're first deaminated, then converted into pyruvic acid, a keto acid intermediate of the Krebs cycle, and the events include transamination, oxidative deamination, and keto acid modification. These are the steps we just actually talked about. During transamination, an amine group is switched from an amino acid to a keto acid. In oxidative deamination, the amine group of a glutamic acid is removed as ammonia and combined with CO2 to form urea. And finally, during keto acid modification, the keto acids formed during transamination are altered so they can easily enter the Krebs cycle pathway. So you can see that process as you go through transamination. Protein synthesis is normally hormonally controlled. It requires a complete set of amino acids, and essentially amino acids must be provided within the diet. So without them, protein synthesis can't occur. You have to have all of them. There's a catabolic anabolic steady state, and it's a dynamic state in which organic molecules, except for DNA, are continuously broken down and rebuilt, and organs have different fuel preferences, so that's why that's so. There are three inconvertible pools. There are amino acids, carbohydrates, and fats. These are the three essential pools of our nutrients. The amino acid pool is the body's total supply of free amino acids, is a source for resynthesizing body proteins, forming amino acid derivatives, and then gluconeogenesis if we have too much. And this takes you through that process of how all those things happen. We have carbohydrate and fat pools. They're easily interconverted through key intermediates. They differ from the amino acid pool in that fats and carbohydrates are actually oxidized directly to produce energy, and excess carbohydrates and fats can be stored, where protein not so much. And this takes you through, again, that process we just talked about. We have absorptive and post-absorptive states. Fed states are during and shortly after eating. The absorption of nutrients is occurring. Post-absorptive or fasting states is when the GI tract is empty and energy sources are supplied by the breakdown of those reserved materials we talked about. The absorptive state, anabolism exceeds catabolism. That is that we're taking in things and we're producing more, we're building up more molecules and we're breaking down. Carbohydrates, glucose is a major energy fuel, and glucose is converted to glycogen or fat during the absorptive states. Fats, the lipoprotein lipase hydrolyzes lipids to chylomicrons in the muscles and fat tissue, and most glycerol and fatty acids are converted to triglycerides for future storage, or for storage for future use. Triglycerides are used by adipose tissue, liver, and skeletal cardiac muscles as a primary source of energy, so that's kind of important to know. Proteins, excess amino acids, are deaminated and used for ATP synthesis, or they're stored as fat in the liver, and most amino acids are really used in protein synthesis. And this takes you through all that stuff that we just talked about, and those are the major events of the absorptive state. Takes you through the principal pathways of absorptive state. The hormonal control of absorptive state, insulin secretion is stimulated by elevated blood levels of glucose and amino acids, and GIP and parasympathetic stimulation. So important to know that that's true. Insulin effects on metabolism. Insulin is a hypoglycemic hormone which enhances facilitated diffusion of glucose in the muscles and adipose tissue where it can be used. Glucose oxidation, glycogen and triglyceride formation, and the active transport of amino acids into tissue cells, and finally, protein synthesis. This takes you all the way through what we just talked about. We have a post-absorptive state. That's where the catabolism of fat, glycogen, and proteins actually exceeds the buildup of those same materials through anabolism. The goal is to maintain blood glucose between meals, and it makes glucose available to the blood. It promotes the use of fats for energy and is glucose sparing, and the goal is to use up the fat. Sources of blood glucose include glyconeogenesis in the liver, glycogenolysis in the skeletal muscles, lipolysis in adipose tissue in the liver, and that's where glycerol is used for gluconeogenesis in the liver. Catabolism of the cellular protein during prolonged fasting, where this is where the source of blood glucose come from amino acids, which are deaminated and used for gluconeogenesis in the liver and later in the kidneys. This takes you again through that process. Again, this takes you to the process as it goes through the body. The post-absorptive state, there are hormonal controls. Glucagon, another hormone released by the pancreas, is released and stimulated by declining blood glucose and rising amino acid levels. The glucagon, which is a hyperglycemic hormone, which raises blood sugar, promotes glycogenolysis and gluconeogenesis in the liver. It promotes lipolysis in adipose tissue, and it promotes modulation of glucose effect after a high-protein, low-carbohydrate meal. This, again, takes you through that whole process that we just talked about. The post-absorptive state, the neural controls, in response to low plasma glucose or during fight or flight or exercise, the sympathetic nervous system and epinephrine from the adrenal medulla promote fat mobilization and glycogenolysis. The metabolic role of the liver is hepatocytes process nearly every class of nutrient. They play a major role in regulating plasma and cholesterol levels. They store vitamins and minerals. They metabolize alcohol, drugs, hormones, and bilirubin, which is a after product of the breakdown of red blood cells. Cholesterol is the structural basis of bile salts, steroid hormones, and also vitamin D. It's the major component of plasma membranes. It makes up part of the hedgehog signaling molecule that directs embryonic development. It's transported in lipoprotein complexes containing triglycerides, phospholipids, cholesterol, and also protein. There are several types of lipoproteins. They come in HDLs, LDLs, and VLDLs, and chylomicrons. HDLs, which are high-density lipoproteins, have the highest protein content. LDLs are low-density proteins, and they are cholesterol-rich, and so that's why your doctor is always trying to get you to lower your LDL. And VLDLs are very low-density lipoproteins, and they're mostly triglycerides. And then the chylomicrons, which are a transport molecule. And this talks about how they're transported across and what percentage of triglyceride, phospholipid, cholesterol, and protein is in each of these types of molecules. Lipoproteins, the VLDLs, transport triglyceride to peripheral tissues, mostly adipose tissue. The LDLs transport cholesterol to peripheral tissues for membrane storage and hormonal synthesis. HDLs transport excess cholesterol from the peripheral tissue to the liver, be broken down and secreted into bile, and they also provide cholesterol to steroid-producing organs, and we'll talk more about that later. High levels of HDL are thought to protect against heart attack, and high levels of LDL, especially lipoprotein, increase the risk of heart attack. So you'd like to have really high HDLs and really low LDLs. Plasma cholesterol levels, the liver produces cholesterol, and at the basal levels, regardless of the dietary cholesterol intake. So a lot of where your cholesterol levels are is really dictated by genetics and what the liver is doing. And that's usually in response to saturated fatty acids. Saturated fatty acids stimulate the liver synthesis of cholesterol. They inhibit cholesterol excretion from the body. Unsaturated fatty acids enhance excretion of cholesterol. So saturated fats are not as good as unsaturated fats. Trans fats increase LDLs and reduce HDL. So trans fats are those fats we can take in from some of our food ingestion. Plasma cholesterol levels, unsaturated omega-3 fatty acids, which are found in cold water fish, lower the proportions of saturated fats and cholesterol. They have an antiarrhythmic effect on the heart, which can stop antiarrhythmias. They help prevent spontaneous clotting, and they also lower blood pressure, so they have a very positive effect on the body. Non-dietary factors affecting cholesterol, stress, cigarette smoking, and coffee actually lower our HDL levels. Aerobic exercise and estrogen increase our HDL levels and decrease our LDL levels. So there's a lot of value in doing aerobic exercise. Body shape is important. There's called the apple shape and the pear shape. Apple shape is where the body's kind of rounded. Fat is carried on the upper body and is correlated with high cholesterol and LDL levels. A pear shape, where the fat is carried on the hips and thighs, is correlated with low cholesterol and LDL levels. So this just explains that. Energy balance. Bond energy is released from food, which must equal the total energy output. So energy released from food must equal total energy output. Energy intake equals the energy liberated during food oxidation. Energy output includes what's immediate loss to heat, which is 60%, strangely enough. It's used to do work, which is driven by ATP, and it can be stored as fat or glycogen. Heat energy cannot be used to do work. It warms the tissues of the blood, helps to maintain the homeostatic body temperature, and allows metabolic functions to occur efficiently. Obesity, that's where the body mass index, BMI, equals the weight times 705 over the height in inches, and you're considered overweight if the BMI is 25 to 30. It's considered obese if the BMI is greater than 30. There's a higher incidence if the BMI is greater than 30 of atherosclerotic heart disease, diabetes mellitus, hypertension, heart disease, and osteoarthritis. The regulation of food intake. There's two distinct sets of hypothalamic neurons, LHA and VMN. LHA neurons promote hunger when stimulated by neuropeptides, and that's IENPY. VMN neurons cause satiety. They make you feel satiated through the release of CRH hormone, which stimulated by appetite suppression peptides, so POMC and CART peptides, if you will. The factors that affect brain thermoreceptors and chemoreceptors are neurosignals for the digestive tract, blood-borne signals which are related to body energy stores, hormones, and to a lesser extent, body temperatures and psychological factors. Neuroscience. High protein content in meals increases and prolongs the afferent vagal signals. Distension sends signals along the vagus nerve that suppresses the hunger center. So if you make your stomach feel full, you get a negative feedback, if you will. So if you eat things that make your stomach feel full but are high in calorie, then you still get the same result. Nutrient signals increase nutrient levels of blood to suppress eating, like blood glucose, amino acids, and fatty acids. Hormones, gut hormones, in other words, insulin and CCK, depress hunger. Glucagon and epinephrine stimulate hunger. And ghrelin, GHR, from the stomach stimulates appetite just before a meal. It makes you think like I'm going to be hungry. Leptin is that hormone which is secreted by fat cells in response to increased body fat mass, and it's an indicator of total energy stores in fat tissue. It protects against weight loss at times of nutritional deprivation. In ancient times or in prehistoric times, man would not always have everything to eat, and so when he had took in too many calories, he would store as fat. And the leptin would cause more fat to be stored until he would have to go into a point where he couldn't eat anymore for long periods of time, and they would use up the fat. Leptin acts on the ARC neurons in the hypothalamus. It suppresses the secretion of NPY, a potent appetite stimulant, and it stimulates the expression of appetite suppressants, i.e. CART peptides. This takes you to that whole process that we just talked about. Long-term regulation of food intake, there are additional factors: temperature, stress, psychological factors, adenovirus infections, and sleep deprivation. Total heat produced by chemical reactions and mechanical work of the body defines our metabolic rate. It's measured directly with a calorimeter or indirectly with a respirometer. The basal metabolic rate, the BMR, reflects the energy the body needs to perform its most essential activities. And factors that influence our BMR, as the ratio of body surface area to volume increases, BMR increases. Our BMR actually decreases with age, but it does increase with temperature or stress, and males have a disproportionately higher BMR than women, and thyroxine increases oxygen consumption and cellular respiration in BMR, and so thyroxine being that thyroid hormone. The metabolic rate, the total metabolic rate, is the rate of kilocalorie consumption of fuel in all ongoing activities. It increases with skeletal muscle activity and food ingestion. Regulation of body temperature. The body temperature reflects the balance between heat production and heat loss. At rest, the liver, the heart, the brain, kidneys, and endocrine organs generate most of the heat with the, if you will, most of the heat. During exercise, heat production for skeletal muscles increases dramatically. The normal body temperature is 37 degrees centigrade or 98.6 degrees Fahrenheit. It provides an optimal environment for enzyme activity, and increased temperature denatures the protein and actually depresses the neurons and inhibits optimal protein activity. This kind of takes you through that process of what happens. Core and shell temperature. Organs in core have the highest temperature. Blood is a major agent of heat exchange between the core and the body shell, and core temperature is regulated. Core temperature remains relatively constant, while the shell temperature can fluctuate a lot depending on the outside environment, whether we're active or not. The mechanisms of heat exchange are four mechanisms: radiation, conduction, convection, or evaporation. Radiation is a loss of heat in the form of infrared rays. Conduction is a transfer of heat by direct contact. For instance, if you lay on a cold cement floor, you conduct your heat to the cement floor. Convection is a transfer of heat to the surrounding air, and evaporation is heat loss due to evaporation of water from the body surfaces, like when you sweat. And this takes you to kind of a nice picture. Boy, don't we all wish we were there. Mechanisms of heat exchange: insensible heat loss accompanies insensible water loss to the lungs, the oral mucosa, and the skin. Evaporative heat loss becomes sensible, active, when the body temperature rises, and sweating increases our production of water vapor. We talk about the role of hypothalamus. The preoptic region of the hypothalamus contains two thermoregulatory centers: the heat loss center and the heat promoting center. The hypothalamus receives afferent input from the peripheral thermal receptors in the skin and the central thermal receptors, some in the hypothalamus. It initiates appropriate heat loss and heat promoting activities within the body, so it manages the heat. It's sort of our thermostat, as it were. Constriction, heat promoting mechanisms: constriction of the cutaneous blood vessels, which causes shivering, and increased metabolic rate via epinephrine and norepinephrine, and enhanced thyroxine release, which actually increases our heat levels. Voluntary measures include putting on more clothing, drinking hot fluids, and changing our posture, increasing our physical activity. Heat loss mechanisms: dilation of the cutaneous blood vessels, or the blood vessels in our skin. We have enhanced sweating, or we sweat. We have voluntary measures, which include reducing our activity and seeking a cooler environment, or wearing light-colored and loose-fitting clothing in high temperatures. And this takes us through that homeostasis process. And way back in Chapter 1, we had done a homeostasis lab, and you can remember how that went. And this takes you all the way through that yet again. Remember that hyperthermia is a condition where there's elevated body temperature, and that depresses the hypothalamus. There's a positive feedback mechanism called a heat stroke, which begins the core temperature at 41 degrees centigrade. And if it's not corrected, it can be fatal. Heat exhaustion is heat-associated collapse after vigorous exercise. It's due to dehydration and low blood pressure. The heat loss mechanisms are still functional, and it could progress to heat stroke if not managed. Hypothermia is really low body temperature where vital signs are decreased. Shivering stops the core temperature of 30 to 32 degrees centigrade, and it can progress to coma or death by cardiac arrest at 21 degrees centigrade. Now, interesting enough, people have been taken down to 60 degrees centigrade because they fell in the cold water and became hypothermic to 60 degrees centigrade, and were brought back with no apparent long-term symptoms. Fever is controlled hyperthermia. Its causes an infection, which could also be cancer, allergies, or injuries of the central nervous system. And it's where macrophages release interleukins, which are called pyrogens. They produce increase in temperature that cause the release of prostaglandins from the hypothalamus. Prostaglandins reset the hypothalamic thermostat to be much higher, and so the natural body defenses or antibiotics reverse the disease process. For instance, cryogens, vasopressin, resets the thermostat to a lower level. So it gives you kind of a feel for what happens during the fever process. All right, this has been the lecture of Chapter 23, recorded lecture Chapter 23 on nutrition, metabolism, and body temperature regulation. Thank you very much.